ঠিক আছে স্যার, আপনি স্টার্ট করে দিন।
আরে আমি করে দিচ্ছি, ঠিক আছে, আমি করে রেকর্ডিং তো থাকবেই, এটা পেয়ে যাবে ইউটিউবে। ইম্পর্টেন্ট ক্লাস ছিল তো বেটার ছিল জয়েন করা উচিত ওদের, জানি না যা পারে। আমাদের তাহলে এই তো কমপ্লিট হয়ে গেছে। টুডে উইল স্টার্ট দ্য নিউ টপিকস, দ্য প্রোটিন সিনথেসিস or translation. Now just doing these slides. Today we start. Good morning everybody, my dear students. The today we start about the translation or the protein synthesis. This is the very important process in the central dogma of life, and this is the last process, the end process of central dogma of life, translation or protein synthesis. Just replication, transcription, post transcription and modification. After that the protein synthesis will be occurred, and if the protein is properly synthesized then it is useful for the physiological function and any biological function. But if it is a faulty preparation of protein, that will be different types of disease or the disorders. So protein synthesis and a actual requirement of the protein synthesis, this is very important. If so many factors can help the protein synthesis, but there is a mischance or misleading of protein synthesis in the so many factors. So be careful, be take care, be learn thoroughly, otherwise it will be a problem. Not only problem in the protein synthesis, problem in your examination also. So my dear student, please remember it carefully. This is the central dogma of life: replication, transcription and translation. The mRNA will be produced by the transcription. This mRNA after translation it produces some protein synthesis. It produces protein. Okay. Cell are governed by the cellular chain of command DNA, RNA and protein. And in case of transcription, it is the protein synthesis of RNA under the direction of DNA and process of messenger RNA. And translation, it is the actual synthesis of the polypeptide, which occurs under the direction of mRNA. That mRNA produced by the transcription or by the instruction of the DNA and occurs in the ribosome, and ribosome is called the protein house, protein synthesis house, and where the two subunit, in case of one subunit, large subunit and only the small subunit in case of prokaryotic, the 70s, and large unit the 50s and 30s, in case of eukaryotic 80s, and the small unit is the 40s and the large unit the 60s, mammals to eukaryotics to mammals, and as well as the human also. So these are the ribosome, and it is the factory house, and the tRNA is the transfer RNA or just like a truck, and mRNA is the messenger RNA, that are the message carry which type of protein is synthesized, and ribosomal RNA, which is the ribosome to initiate the protein synthesis in the ribosomes, and there is some initiation factors are involved in this protein synthesis. The transcription and translation, in case of prokaryotic, transcription and translation occurs together, but in case of eukaryotic it is a most complicated method. After transcription then the protein synthesis or translation will be occurs. Transcription and translation in case of eukaryotic cell are the nucleus envelope separated transcription from the translation and intrinsic trans RNA process in which the nucleus, and the nucleus with the translation DNA, pre-RNA, mRNA and the ribosome polypeptide will be formed. In case of nucleotide cells, the nucleotide provides the separate customs compartment of the translation. It is originated RNA transcription called the pre-mRNA, and its process is in various ways before leaving the nucleus as mRNA. So I think this is the another slides better to understand. The translation. Translation is the language of nucleotide sequence on mRNA or genetic code. Last class we discussed about the thorough about the genetic code is translated into the language of amino acid. Actually this genetic code, the triplet code, which are the sequence and the sequence which correlate or translate into the amino acid code, and The polymer of amino acid is nothing but a polypeptides or the protein, and which is essential for our body, for the building blocker of our body, and the so many function of our body. Now, the tRNA, it is the most important component for protein synthesis. tRNA, or the soluble protein, or just like a track, or here it is called the transfer RNA. It transfers the amino acid from body cell to ribosome and transfer or AA form in the cytosol to form a ribosomal protein synthesis machinery, around 73 to 93 nucleotides in the length. And here is the just like a single strand molecule of tRNA, just like a mRNA, but the structure is the different, quite different than the mRNA. But here it is the two arms, one is the DHU arm, another the tau or C arms, another the N ending, one is the amino acid binding side, another is the polypeptide binding side, another is the anticodon site. So given in the picture in the details in the next slides, this is the code for the mRNA 5 prime to 3 primes. Here it is a, this is the code, just it will be reversed. Here you can see this amino acid acceptor arm, here the arms 3 prime arms where the amino acid can binds, and this is the ribosomal binding site. When that means which ribosome the large or small subunit that will be bind, these are the tau T tau C arms, another the DH arm, the TRA molecule, which is recognized the specific enzyme amino acid transferase, which or D arms, which bind with the amino acid with the specific enzyme amino acid tRNA and tRNA. This anticodon GCC, which is the codon for 5 prime to always 3 prime reaction. This is the antimolecular base pairing and the anticodon. This anticodon site which bind with the mRNA. So these are the anticodon and this is the mRNA sequence. The depend upon the mRNA sequence which type of tRNA bind, this is depend upon the anticodon of this particular type, and simultaneously this amino acid must be carried. So in case of the ribosomes, the interstructure for the mRNA and tRNA and amino acid to enter into the each of other translation. This is the basic component for the protein synthesis. One is the tRNA, mRNA, RNA, protein, amino acid tRNA synthesis. So many factors, and 80S in case of eukaryotic, 60S prokaryotic, and 60S large, 60S in case of 50S in case of subunit, in case of small, 40S in case of eukaryotic, and 30S in case of prokaryotics. Requirement of the translation: first is the ribosome, because it is the factory house for the protein synthesis. Another molecule mRNA, which carry the message which type of protein is synthesized. Next is the tRNA, which is transfer the amino acid, or it is like a track and amino acids, the amino acid molecule, which amino acid after say formation of polypeptide, which amino acid or what type of amino acid. So in that case, when the protein synthesis will occur, all type of amino acid must be present inside the cell, otherwise it will be a faulty preparation of amino polypeptide. So large number of or adequate amount of amino acid must be present in the cell, otherwise the difficult to protein synthesis or difficult to different types of protein sequence. If the sequence is different, there is a different types of protein will be formed, so they are biologically either inactive or active. The amino acid tRNA synthesis, another enzyme ATP, which is the energy ATP, and GTP, which is essential for the supply for the energy for the protein synthesis. So it is a energy dependent process, and enzymes of the translation and the protein factor. So many protein factor or initiation factor for synthesis of the protein. In every function will be factorized by the initiation factors. Translation step: first is the activation of the amino acid, then initiation, then elongation, termination, and post translation modification. So activation of amino acid, we can start the amino acyl tRNA synthesis. Amino acid plus tRNA plus ATP equal to amino acyl tRNA synthesis and AMP. Activation of amino acid tRNA, it is the D arm of the tRNA are recognized by the enzymes amino acids tRNA and ATP amino acyl tRNA and AMP. Enzymes defines the specific tRNA and transfer the amino acyl group to 3N CCA, and this COOH group of amino acid is reactified with the 3H group of tRNA and at least enzymes for one amino acids. So amino acid, how it will be the activation? Then you can see it. This is the amino acid with amino acyl tRNA synthase, ATP to PPI and 2 PPI, 2PI, ultimately EMP, adenosine monophosphate amino acid. This AMP with CCA bind with the AMP, E will be released, and this CCA amino acid bind with the this 3' M and amino acid tRNA will be produced. This attachment of specific amino acid to the corresponding tRNA by the amino acid tRNA synthase E enzymes. That means the different types of enzymes bind with the different types of tRNA. At least 50 different types of tRNA is available in our cell, and 20 amino acid bind with the tRNA as per the anticodon bind with the mRNA. So tRNA is a very essential component, or is just like a truck to carry out the amino acid from the cells to the mRNA. For example, in the serine, CC which bind with the anticodon site, देखो AGU, it is the anticodon, and this is the messenger RNA UCG, their anticodon AGU, it is a code for serine. If you see it, the complete genetic code EAGU, it is a code for serine, and that serine will be attached with the amino acid. That means this tRNA carry the serine amino acid, and which is bind with the anticodon of mRNA. And as per traditional base pairing rule, they are produced. That is called the Hubble hypothesis. I discussed in the previous classes. This mRNA is read 5' to 3' direction, and protein synthesis takes place. And from a N terminal to C terminals, that means amino terminal to carboxylic terminals. How it will be initiation? This is the very good important factors of initiation. How it will be initiated? Any work can be done by the some initiation factors. Suppose say for example any social work or any class or any events, there is some initiation, some student are initiative, after that some student are joined here, and finally the work will be done. Otherwise it will be possible. So their initiation is very important. একটা বাংলায় কথা আছে বিড়ালের গোড়ায় ঘন্টা বাঁধবে কে, because the initiation factor, which is a very essential component, who start the protein synthesis or who start the any events, this is the very important. After that so many students or so many people are come, but who started the or who initiated, this is very important. Ribosome, one is the key factors, that is the protein synthesis mRNA to translate, and the initiation of met tRNA. This met tRNA methyl tRNA, that means basically it is for, or eventually it is occur in the prokaryotics, not in case of eukaryotics. A set of protein factors called the initiation factors, and in case of eukaryotics there are at least 9 factors designated as a initiation factors. If EF, that indicates the eukaryotic origins. In prokaryotes, three initiation factors are known as the initiation factor 1 and initiation factor 2 and initiation factor 2. In case of 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 prokaryotic 3 factors, or in case of eukaryotics it is 9 factors for protein synthesis. Next steps involved in the initiation: ribosomal dissociation, formation of the 43S for the pre-initiation complex, formation of 48S initiation complex, and formation of 80S initiation complex. Ribosomal dissociation for ILF 3 initiation factor 3, initiation factor 1 and 40S subunit, and prevents the association with the 50S causing the ribosomal subunit dissociation. Formation 40, this is not required. Then the formation of 80S ribosome or ELF factors. This is the 30S ribosomal factors or ribosome dissociation, 50S and 40S. Here eukaryotic will be seen, initiation factor 3, initiation factor 2. This tunnel mate UCC that bind with this. The 30S pre-initiation, this is These are the first step of reaction in the met arginine, which is CGCU and CGU. It is formation of this type of reaction, and AUG, it is the start codon for protein synthesis, and it will be formed UC, UAC, UC. This is amino acid binding site. Here is the peptide binding site. The amino acid will be bind one amino acid attached with the then another amino acids. The elongation, the binding of the new amino acid tRNA and code for the mRNA determines in the hetero coming on the EFA and the groups, and the peptide bonds is formed. So this NH2 group is incoming into the amino acid A site, there will be an amino acid binding site, and steps a peptide forms to form a groups to form a peptide site, and enzyme peptidate transferase. The amino acid tRNA transferase, which is responsible for the amino acid bind to the tRNA, and peptidate transferase, which is bind with the amino acid to form a peptide bond and to form a polypeptides and to form a particularly the protein. The translocator with the peptide bonds and the formed of transfer protein shifted to site and tRNA. It is stepped and formed sites. Now the P sites are free. Ribosome moves from towards one codon to peptidal tRNA and transparent tRNA sites. Now at the site of empty, the new amino acid tRNA will commonly to a site, and the steps to required F2 and energy form and GTP, and this new amino acid or tRNA can confine elongation to continue. I think this is the elongation. I think this initiation part will be today. After that, elongation and termination, how it will be terminated in the next class. If you have any question, please ask me or any. Thank you very much for listening the lecture.